Today is going to be about bioinformaticians and genome analysts, who we are and what we do and how we build these complex genomics pipelines. You've heard about how the field is advancing in terms of the technology and also in terms of the knowledge around the clinical application of that knowledge, but how do we go from one to the other? And so the first part will be talking about who we are and what we do, and the second part will be more about how we approach interpreting this complex data. So this is just a disclaimer to say that I do work on the POG project, so all of my examples were based around the POG examples, but the principles that I'm outlining do apply to other genomics platforms as well. And I'm talking in more of the context as the field moves towards to more complex levels of information and interlaying multiple types of information, that how do we start to deal with this complex array of information to give you something that is actually digestible and usable in the clinic. So the need, which is still, this is the constantly evolving field, so it's still an incompletely met need. So the data itself, genomics data, is very complex. You don't really need to know what this figure is trying to show, except that this is one data set of the genomic information, and some talented data analyst has tried to resolve some information from this complex data set. And you can see that even just mapping these things out in a visual representation can be very challenging. Also, the data is massive. There's lots of it. In terms of multi-omics approaches, we're starting to get lots of information. So this image here is showing a genome arrayed in a circular form, the chromosomes in a circus plot. But every layer inside that circle is a different type of information from one patient. So we have structural variants, small nucleotide variants, copy number changes, expression data. So integrating all of this together to get a complete picture of what's going on in the tumor is very complex and also very important. The data is imperfect, so for various reasons. There might be gaps in the data, particularly in terms of sample quality. Sometimes things are missing. Also in computational and technical terms, the data might come through incomplete, so you do need to have checks and balances in place to manage that data to make sure that you're working with high quality and informative information. And then finally, going back to Stephen's fruit analogy, the field's constantly evolving, so we have to be adaptable to be able to integrate new emerging technologies that can be informative in the clinical sphere as well. So to do this, we need developers, curators, translators, all to manage and interpret this data. So that's broadly what bioinformaticians are or evolving to be. But just a note on the specializations that, when back in the day when genomics was relatively simple compared to what it is now, a bioinformatician was a broad catch-all for someone who could take a pipeline from start to finish through building the infrastructure to developing. So this is nothing new. We've been doing this for millennia in terms of industrial processes, but it's just good to be aware that now we're starting to evolve into specializations within the bioinformatics field, where you'll have people who are only directed towards building infrastructure rather than understanding and analyzing the data at a genomic level or a biological level, and then genome analysts who are more at the other end of the spectrum, understanding the biology and the informatics that's gone to creating that information. And of course, bringing this all into the clinic, it brings in you guys, the clinicians, the pathologists, and other in the clinical sphere. And so now this is really the point I'm making, is that this is a interdisciplinary effort. So all of these different groups that are stratified throughout the process need to understand what each other's doing and help to inform each other as we develop these pipelines. Background is who can use mathematics and information theory to organize this complex and biological data. After those two activities have gone on, the generating the data and generating the knowledge that can be used to interpret the data, you need someone to actually go and do that interpretation, and that's the activity that's being filled by the genome analyst. And so this is a person who has a broad understanding of the bioinformatics background that goes into creating the data, as well as a cancer genomics background. So, because it's really important to understand where the data's come from, how reliable is the data, and because that changes how you understand and interpret the data. So this is an image that I'm going to come back to a few times in the talk. It's just a typical workflow. This represents what we do in POG quite well, but also other genomics platforms, where you go from basically producing the data to a process of filtering, annotating, so you get to a high-quality data set where some human individual can go and look at that data and start to interpret and understand what it means. And then once they've put their filter of perhaps knowledge and expertise onto that, to then pass that on to the clinical activity. And also, while all this is going on, we have to keep an eye on quality control, the throughput, the turnaround times, as Stephen was alluding to, the reproducibility of the data so that we are confident in what the data we're showing, as well as being adaptable to the changes in the field as well. And so we can break this down for us in terms of 4 main activities that go on. So pipeline development and building the infrastructure is a key activity. You can't do anything else without having a good pipeline. But one of the most important aspects as we move into the clinical sphere with genomics technology is acquiring the relevant information that we need to apply to the genomics information. And of course, then combining these 2 activities into analyzing and interpreting that data so we can pass that on to communicating to the clinician. One of the most important activities that we're currently developing and is relatively new to the field, and is quite a complex issue, is this issue of collating the domain information in a usable way. So we need experts who have domain knowledge in terms of biology and in terms of the clinical genomics to be able to organize and manage this information in a relevant way. And so to do that, the general term that's coming out is building a knowledge base. So this is essentially a database of information that's relevant to your pipeline. So we know all the information in the world is available on the internet, but it's not easily accessible. So what we're doing here is we're trying to stay current with the rapidly evolving field and trying to capture the relevant information, such as therapeutic, diagnostic, prognostic, and biological information, and capture this in a state that can be then computationally queried so we can start to do automatic associations between patient data and knowledge and produce automated reports that don't require a lot of hands-on activity. Okay, so that's just a summary of some of the activities that go into these pipelines. I just want to run through the second part of the talk, which is how to approach interpreting the data itself. And this is going to be fairly brief. But we've heard a lot from Marco and Stephen about the different types of data that's available, so genome, epigenome, transcriptome, proteomic, all of this different levels of information build up. And the important point to make is that these actually give you unique information. It's not like you could say we could get all the information from one type of approach necessarily. Layering this information together can be very fundamentally informative. And so, but it is important to understand how this data was produced and the different sensitivities and detection levels between those. So, for example, something like DNA, we can think of as quite well structured, quite a robust data source. So you can think of it almost in digital terms. It's a bit of an oversimplification, perhaps, but in terms of whether you have a variant or you don't, it's a kind of all or nothing signal. So if you have a low tumor content sample, as the tumor content goes down, that signal stays fairly robust, even at 20% tumor content, such as that. Whereas something like expression data from transcriptome is a very complex snapshot of what's going on in the sample. And so there's a lot of interplay between gene expression. There's a lot of different rates in terms of whether genes are turned over rapidly. So this is essentially more of an analog type signal. So as something like tumor content is reduced, that signal can be disrupted more readily than DNA. So when you have a low tumor content sample, for example, you have to be aware of the different weights you put on the information, the different kind of granularity in terms of the interpretation you can get from that. And then just finally, the concept of integrative analysis, whereby we have, say, an AKT mTOR pathway, and you overlay the DNA mutations, you get some knowledge of how this pathway is being perturbed and perhaps how you might action it. But then these are complex pathways. They have a lot of consequences downstream and upstream. So when we overlay expression data, you might see that actually it's this, of the downstream signals, it's maybe these players that are more relevant than something like this over here or these over here, and so that might change your the kind of drugs and the way you want to treat that specific variant, but also overlaying, proteomic data to give you an understanding of how active some of these upstream receptors are, can help you understand which pathways are being active, as well as understanding parallel pathway activation and how those might interact together. So all of this comes together to give you a picture of what's the most relevant axis of target. And of course, the clinical information is very important as well. If you have a mutation in the mTOR pathway, and we're suggesting that you use an mTOR inhibitor, but your patient has already failed to respond to mTOR inhibitors, that's not necessarily the kind of information that you want. So we can integrate that into the analysis to get a more stratified set of options for the patient.